morning, everyone. Today, before I start my presentation, I want to ask you to look at the two pictures. On the first picture, we have two fishes of the same age, but as we can see, the first fish is approximately 3 times greater than the first one. So, and the second picture is the glow-in-the-dark cats. As its name says, these cats glow in the dark. So, what do you think is the relationship between these two pictures? Could you brainstorm? Yeah, that's correct. The relationship between these pictures is the fact that organisms on these pictures were genetically modified. So today my aim is to inform you about the process of genetic modification. Actually, the majority of us is acquainted with the term of genetic engineering, but none of us know the specific process of how genetic modification actually occurs. So the reason I'm explaining this topic today is very simple, that we as a consumer should know how our products, genetically modified products, are produced. So I will try. Actually, the process of genetic engineering is not very so simple, so I'll try to explain it in a clear and easier way. So before I start my presentation, I want preview. In my presentation, I'll cover 4 main steps of genetic engineering. The first one is the DNA cleavage and isolation. The second one is the creation of recombinant DNA. Then is the cloning, and finally screening. So before I move on to my main points, I want to provide a little bit background information. What is genetic engineering and what is the aim of it? So genetic engineering is the modification of an organism's genetic composition by artificial means, which means that we take the gene that codes for one trait from one organism and insert it to another organism with the aim of creating new classes of plant or new breed of an animal. So the first, as we can see, the process of today's topic is the genetic engineering. So the first step of genetic engineering is the DNA cleavage and isolation. This step is done with the help of enzyme that is called restriction endonuclease. So according to the Biopharmaceutical Center, restriction enzymes are proteins that act like scissors and cut this double-stranded DNA into short fragments. So as long as we obtain these DNA fragments, biologists do electrophoresis step. Electrophoresis step is the biological procedure that separates biological molecules, charged biological molecules, into the according to their size. So DNA molecule is actually negatively charged, so which means that DNA moves from the negative electrode to the positive electrode, and it is further stated in Biopharmaceutical Center that the shorter the DNA fragment, the far it will travel, which means that the most short fragments will be here and the most long will be here. And as long as biologists obtain the needed DNA fragment, they extract it from electrophoresis, and then they further use in the next step, which is the creation of the recombinant DNA. So DNA fragment, which is inserted here, is then inserted into a plasmid. A plasmid, according to the National Health Museum website, is the type of DNA that is found outside of nucleus of bacterial cell. In this case, plasmid acts like a vector, which is, according to the Professor Chandler, acts like a vector, or in other words, is a DNA carrier. So then, this DNA fragment obtained from the previous step becomes added to the vector with the help of enzyme called ligase. And this addition of the DNA fragment with the vector creates a recombinant DNA. This recombinant DNA then should to be inserted into a bacterial cell with the help of step which is called electrophoresis. Electrophoresis is actually the same step as electrophoration, but here we again use electric current in order to create pores in the bacterial cell membrane. We create pores in here, and then with the help of electric current, this recombinant DNA becomes inserted into the bacterial cell. So after we obtain a bacteria with needed recombinant DNA, we should increase it in the amount, in number. That's why we use the third step is the cloning. The cloning is the step is a procedure when we provide favorable conditions for bacteria where it can reproduce. According to the MIT University, one bacteria can divide each 20 minutes, which means that in a period of overnight, biologists can obtain millions of these bacteria. But however, none of these bacteria will obtain the needed recombinant DNA, which means that we should check does this bacteria possess the recombinant DNA that we needed or not. So the last step for checking is screening. According to the Life Technology website, there are 5 strategies of checking or screening bacteria. And all of them actually are based on the usage of a special chemical that will allow the growth of only bacteria that possess this recombinant DNA. So, as a result of this 4th step, biologists obtain a bacterial cell with recombinant DNA. With the help of these bacterial cells, biologists can then obtain new breeds and new classes of plants. So, to sum up, during my presentation, I tried to explain you the process of genetic engineering. Namely, I covered the 4 main steps. The first one is the DNA cleavage and isolation. Secondly, I covered the creating a recombinant DNA by inserting into a bacterial cell. Then I covered the cloning of genetic engineering, and the final is the checking these cloned bacteria, which is screening. So my aim was to inform you about the process of how genetically modified products are actually made that we are already consuming. We as consumers should know how these products are made. I was not here to persuade you to consume genetically modified products or not. I was here to inform you how genetically engineered products are actually made. So thank you for your attention.